Okay, so today I'm going to talk about how Calvin figured out all the different carbon conversions that happen in a plant during the process of photosynthesis. So he basically did this through an experiment called the Lollipop experiment. And the reason it's called that is just that the glassware or the apparatus that he used is shaped like an upside down lollipop, as you can see in this diagram here. And just a note, none of these diagrams that I mentioned in this video need to be memorized. It's just so that you can see what I'm talking about. So let's break down the different steps of the Lollipop experiment. Firstly, radioactive carbon-14 is added to the Lollipop apparatus containing this green algae, which is basically the plant which will be photosynthesizing and which will be tested in this experiment. Again, it's radioactive just so that it's like a marker. We can actually see where it is and measure it. So light is shone onto the apparatus to induce photosynthesis, which incorporates this carbon-14 into the organic compounds that are produced. So any glucose that this algae produces will now contain this radioactive carbon-14. And again, the equation for photosynthesis involves water and carbon dioxide to make glucose and oxygen. So that's why we need this air and carbon dioxide to be pumped in, because the air does contain water vapor. And then the air outlet is just where the oxygen and anything that's not being used in the reaction comes out. So after a set amount of time, the algae is put into this solution of heated alcohol. And this basically kills the plant by preventing cell metabolism in it. And the algae actually does need to be dead so that we can analyze it. So these dead algae samples are now analyzed using 2D chromatography. So basically, as you can see on this diagram here, the substance begins to separate into many different photosynthetic carbon compounds. And this is done again here. So it's basically done twice, and this is what we call chromatography in 2 dimensions. So this is basically just like a bit rotated, if you can tell, like this is basically the same thing as this. And this just starts to separate. Again, it needs to be in some solvent. So these radioactive compounds on the chromatogram are then identified using autoradiography, which is basically just X-ray film exposure. And it basically produces these different images which can be compared. So by comparing these different time periods of light exposure, the order by which carbon compounds are generated can be determined. So at 5 seconds, ribulose biphosphate is present. At 15 seconds, we have glycerate 3-phosphate, and at 30 seconds, we have triose phosphate. So basically, Calvin used this information to propose a sequence of events known as the Calvin cycle, or the light-independent reactions involved in photosynthesis. So if you look at the Calvin cycle diagram, you'll see that it actually does fit the structure. It starts with ribulose biphosphate, which is then converted into glycerate 3-phosphate, which is then converted into triose phosphate. And this is just a cycle that keeps going. So yeah, that's the Lollipop experiment. If you would like to learn more about the Calvin cycle itself in more detail, we've already made a video on that, so do check it out. Thank you so much for listening, and I hope this video was helpful.